AS Level Biology 9700, Paper 1, Variant 3, October, November 2025. And in this video, we are going to discuss question number 1 to 13. Now, question 1 is on a diagram. The diagram shows a stage micrometer with division 0.1 millimeter apart, viewed through an eyepiece containing a graticule. So the same eyepiece is now used to examine a blood smear. How many graticule units will cover the diameter of a lymphocyte of 10 micrometer? So here you can see that the upper one is the stage micrometer, and the lower is eyepiece graticule. So first of all, you can see the stage micrometer division distance are 0.1 millimeter, which has been given, and lymphocyte diameter, which we has to find out, is about 10 micrometer. So this is our target, that how we will have to find the diameter of a lymphocyte of 10 micrometer. Now we have to calibrate the eyepiece graticule to find out the correct option. So the first large vertical line you can see that is on 10, and the other is on 50, and the third one is on 90. So the distance between 10 and 40 and 50 to 90 is about 40. So we can say that this is the, if we minus 50 minus 10 is equal to 40 graticule units, divisions or units as well. So actual distance on stage micrometer is about 40 graticule units is equal to 0.1 millimeter. So we will convert the units into micrometer. So for this purpose, we will convert the 0.1 millimeter into micrometer by multiplying it with 1000. So it will become 100 micrometer. Now 40 graticule units are divisions are 100 millimeter. So now we have to calculate the value of 1 graticule unit. So the 1 graticule unit or division will be equal to 100 by 40. So it is equal to 2.5 micrometer. So 1 graticule unit is equal to 2.5 micrometer. Now we have the find out the number of divisions for a 10 micrometer lymphocytes. So the formula will be over here. I will write over here number of divisions is equal to size of lymphocytes divided by value of 1 division. So that is equal to, the size of the lymphocyte is 10 micrometer. So it will be 10 divided by value of 1 division, which is 2.5 micrometer. So it will be equal to 4. So after this calculation, we have came to know that the answer is B. So the B option is the correct option. A, C, and D are not the correct options as we have calculated this answer from the given values. Now the question number 2 is structures can be isolated by centrifuge the cells in a sucrose density gradient. This breaks up the cells and separates the cell structures into fractions. So the diagram shows the positions of some of these fractions in a sucrose density gradient. Now the fraction 1 is ribosomes, fraction 2 is lysosomes, and fraction 3 is mitochondria, and fraction 4 is chloroplast. Which fraction contains cell structures that carry out protein synthesis? Ribosomes, definitely they do the process of photosynthesis, or they are the primary sites of translation. And second, lysosomes. Lysosomes are the hydrolytic enzymes for intercellular digestion, so they do not synthesize protein. Mitochondria, yes, they contains their own 70S ribosomes and DNA to synthesize some of their own mitochondrial proteins. And at number 4, fraction 4 is chloroplast. So like mitochondria, chloroplasts are semi-autonomous organelles containing 70S ribosomes to carry out protein synthesis for chloroplast. So fraction 1, fraction 3, and fraction 4. So according to this, the fraction 1, 3, and 4, the B option is the correct option. A, C, and D are not the correct options. Now moving towards the question number 3. And the question number 3 is there. The diagram shows a plant cell. Now you can see at number 1, number is number 1, number 2, number 3, and number 4. So here you can see at number 4 is chloroplast, and number 2 is the rough endoplasmic reticulum, and at number 4 is mitochondria. Now, which labeled organelles would contain nucleotides? Chloroplast. The function of these, the chloroplast, they have semi-autonomous with its own DNA, RNA, and internal nucleotide pools, whereas the Golgi bodies do not have their own genetic material. And number 3, rough endoplasmic reticulum. As you know, the rough endoplasmic reticulum, they have ribosomes attached on them. So you can say they have contained the nucleotides. And whereas the mitochondria, they contain their own DNA, RNA, so they also contain. So according to this, 1, 3, and 4. 1, 3, and 4. C option is the correct option. Now question number 4 is here, and question number 4 shows an image using a microscope. So how many statements about this image are correct? It is an electron micrograph. Yes, it is an electron micrograph because it is uh, highly magnified view of the chloroplast, which is internal um, structure is also visible. It shows part of a eukaryotic cell. Yes, eukaryotic cells has membrane-bound organelles. So this is also correct. It shows at least one mitochondrion. This is chloroplast, not the mitochondrions. So this is not correct. Then it shows a specimen viewed at more than 400 magnification. So this is also correct because it has. So according to this, three statements are correct about this image. So C is the correct option. Now moving towards the question number 5, and you can see over there the question number 5. A bacterial ribosomes consist of two subunits and a total of three. Two subunits, total of three RNA molecules per ribosomes. Three RNA molecules per ribosomes, and analysis of all the 70S ribosomes from a single cell of the bacterium Escherichia coli showed that there were 57,000 RNA molecules. 57,000 RNA molecules, three types of RNA molecules. So 19,000 copies of each type of RNA molecule. So how many 70S ribosomes were there in the E. coli cell? So we have to calculate this step by step. We are told that each individual 70S ribosomes contain exactly three RNA molecules per ribosome. So one copy of each of the three types, we can calculate the total number of ribosomes using either the total number of RNA molecules or the copies per type. So using total RNA type, that is one copy of each type of RNA, and there are 19,000 copies of each type. So there can only be maximum of 19,000 fully formed ribosomes. So B is the correct option. Because there are three, we will divide the total number of RNAs on three, and then we will get the answer 19,000. So they will be equal to this. So this is the correct option. Now the next question is, radioactivity labeled amino acids are introduced into tracheal cell that uses them to make mucus and glycoprotein. So they have given us the number 1, 2, 3, 4 labelings, which root will be amino acid stakes. So for this purpose, which root will be amino acid. So here you can see the this process will take place first cytoplasm. So this uh, you can see. So it will start right from here. It will be at number 1, and then what will happen? You know, this process is taking place in the cytoplasm. So around the nucleus, you can see the rough endoplasmic reticulum are there. On rough endoplasmic reticulum, there are ribosomes attached. So at number 4 will be at number 2, and then after this, the excretory vesicles, which are at number 3. So number 3, they are endoplasmic Golgi apparatus. Golgi apparatus. and this is cytoplasm, and at so it will be at number 3, and at number 4 are secretory vesicles. So the root will be in this, the whole, it is taking place in the cytoplasm. So in the cytoplasm, you will know that rough endoplasmic reticulum, ribosomes are attached on them. So the um, amino acids will go towards they are attached over there. And then the Golgi bodies, apparatus, and then the secretory vesicles. So this will root will be 1, 4, 3, and 2. 1, 4, 3, and 2. So the root will be in this way. As you know, in the cytoplasm, the amino acids are assembled into protein. Cytoplasm, amino acids are assembled into the proteins at the rough endoplasmic reticulum. Then the proteins travel via uh, transport vesicles to Golgi bodies, and from Golgi bodies they will be the secretory vesicles, which will release their contents via exocytosis as at the apical surface of the cell. So in this way, the route will be followed. So B is the correct option over here. Now the next question is question number 7. And question number 7, you can see which features shown in the diagram can be present in viruses. Uh, in viruses, DNA is present and they have protein code, and number 3 is 70S ribosomes. Viruses lack this 70S ribosomes. So they have their RNA. Because some of the viruses contain DNA or RNA, protein code is always there. So you can see that the 1, 2, and 4. 1, 2, and 4 is the B option. So B option is correct. A, C, and D are not the correct options. Now we are going to move towards the question number 8. And question number 8 is the diagram shows some steps in a laboratory method used to identify the presence of non-reducing sugars in a food sample. Some terms are missing. Step X, step Y, and end color C. So which terms are correct for the expected results for a very low concentration of non-reducing sugar? You know, there is a, when we perform the non-reducing sugar test, then at that time, at the end of there, we have to perform the Benedict test. So the Benedict test shows how much of the uh, reducing sugar was present over there, how much of the non-reducing sugar has been broken down. So the color pattern will show the presence of the sugar. So it it starts with blue and green, and then orange, yellow, from yellow to orange, from orange to brick red. It means green carry the lowest amount. If the reaction stopped at green, it means it carries lowest amount of sugar. Yellow more than green, orange more than yellow, and brick red highest amount. So in this way, we can come to know which one is the answer. So at X is the acid hydrolysis, then Benedict test, and then it will be orange contains highest amount. So This cannot be the correct. So according to this, I would like to say first two A and B options cannot be correct. Green, grain option is correct. So, but first of all, we have to perform the acid hydrolysis to break down the non-reducing sugar into reducing sugar. So, acid it has added into it, which will hydrolyze the non-reducing sugar into reducing sugar. So, the first is the acid hydrolysis, then we will conduct the Benedict test when it will be changed into reducing sugar. So according to this, C is the correct option. D is not the correct option. So C is the answer. Next question is, many biological molecules exist as polymers made up of monomer subunits. Which row correctly identifies the monomers in each of the biological molecules? Monomers in each of the biological molecules. Uh, glycogens are alpha glucose, and cellulose are beta glucose, and collagen. This is a protein which is made up of amino acids. So all other three options are not correct. A is the correct option because collagen is protein and it is made up of amino acids. Many monomers are amino acids, and glycogen polysaccharide. So it is highly branched polymer, which is composed of alpha glucose and cellulose structural polysaccharide. So it is found in the cell wall. So it is straight chain polymer made of alternating beta glucose units. So according to this, A is the correct option. Now question number 10 is there. And question number 10 is, which statement correctly explains the result when the two molecules shown in the diagram react together? You can see water has been added to them. So the hydrolyzed reaction is forming the disaccharide maltose. So here you can see the diagram shows two single hexose sugar rings joining together to form a disaccharide with the release of a water molecule. So reaction where molecules join together with the removal of water is a condensation reaction. And where the water is removed and where the water is added, so this is the hydrolyzing reaction. So here you can see hydrolyzed reaction is forming the disaccharide maltose. This is not the correct option. So reducing sugar is split into two monosaccharides by hydrolysis. So they will break down into two monosaccharide by hydrolysis when water will be added into them. So it can be the correct option. Condensation, this is not condensation because water is again, D is showing the condensation is forming. No, condensation is actually the formation by the removal of water molecules, whereas this is the hydrolysis. So our reducing sugar is split by adding the water into two monosaccharides into hydrolysis. So according to this, B is the correct option. Now the question number 11 is there. Which row describes amylopectin? Amylopectin, uh, it consists of alpha 1,4 glycosidic bonds And because it is a branched structure. So branch structure, so many branches are formed over here. So alpha 1,4 glycosidic bond should also is also there. So at 1,4 branch, which is formed 1,4 glycosidic bonds, there is side branch rises which is formed by 1,6 glycosidic bonds. So you can see the shape of the molecule is branched because you can see the different types of glycosidic bonds are formed in the straight chain and in the side chain. So in this way, it will be highly branched. So the function of this amylopectin is the storage. You can see its biological function is to plant's storage as in the form of glucose or energy. So this is the, all these are the present over there. So A is the, according to this, A is the correct option. B, C, and D are not the correct options. Now question number 12. What is a feature of triglyceride molecules? They are hydrophobic molecules. They are molecules with a single ester bond. They are polar molecules. They are water soluble molecules. They are not soluble in water. They are not polar molecules. They are non-polar. They are not polar. So this is not correct. They are molecules with a single ester bond. They contain three ester bond. So this is also not correct. So they are hydrophobic. They are water haters. So A is the correct option because they are not dissolved into, they are not water soluble. So they are hydrophobic molecules. So A is the correct option. An unsaturated fatty acid molecule has 17 carbon atoms in the tail. 17 carbon atoms in the tail, which is dashed to the carboxyl group. The tail contains three double bonds. How many hydrogen atoms does the tail contain? So 17 carbon atoms are there. So fully saturated tail would have each carbon to carbon double bond. So two hydrogen atoms from the structure. Since the tail has three double bonds, we subtract 6 hydrogens from the saturated total number of bonds. So in this way, we can say that, so I would like to write over here, number of hydrogens, that is 2n plus 1. So the given tail n is equal to 17. So 2, 17 plus 1 is equal to 34 plus 1 is equal to 35. So the count for the double bonds, the double bonds have, there are three double bonds. So there are three double bonds. So uh, 3 into 2, they are 6. So we will minus the final total number of hydrogen lost. So the final hydrogen atoms, 35 minus 6 is equal to 29. So according to this, A is the correct option. So it was all about question number 1 and question number 13. So please like, share, and subscribe my channel to watch more videos on this paper. And thanks for watching.